I'm Pratyush Pandey from Educational Series. Today I have come with a new topic, Class 11 Cell Division. I'm in Class 7C and let's start the chapter in a high note. So, cell division. If you have not seen my earlier video which I made up of components of cell and what is cell, so if we see that video, and this is the second part of that video, and this part is divided into two topics: introduction and mitosis, and then another part which will come for meiosis. So let's start the first part, mitosis. Before moving on, please like to subscribe my channel and don't forget to click the bell icon below to get regular dates of my videos. Also, I like to tell you about that this video is a part of Aryabhatta series, which you will get high quality good videos every 1 or 2 in a week. So, cell division. Now we learned in the earlier video that what are cells. Now I told you that cell need to divide to make on their generation or to replace the dead or replace the injured ones. So or for the growth, which is very visual, as when you were 5 years old and till now haven't you grown? Yes, you have grown. So that is growth, not because of every individual cell in our body has grown, but the number of cell has increased itself. It divided, it's increased, the number of cells increased, which caused your body to grow. So in this video we learn about how cell divides. So now this is all why all the cells reproduce by dividing. First they divide all the internal structures, the DNA, and they divide into two, and then using the process of cytokinesis and some the PMAT processes, they divide into two daughter cells. And this you can see this a very good GIF. You can see that the cell duplicates its all parts, DNA, RNA, and then its cytoplasm divides into two daughter cells. As you can see, the other daughter cell go away, and from the parent one stays, and after some time that daughter cell and this parent cell will again divide to make more new cells. Let's proceed on. Cell cycle. Now, as we know our cells, and their cells have a lifetime. So now what do they do all the time? To answer that question, that what are our cells doing at this right moment? Are they watching TV, or are they eating? Are they listening? What are they doing? So to know that part of what our body cells are doing at the right moment is what we learn here in the cell cycle. Cell cycle is the one of the, we can say, the list of daily chores or the work of a cell. This contains some segments about how the cell does its work and how it completes its life cycle or the life cycle of the cell. So now let's see how much time it spends on daily works, growth, replicating its DNA, and dividing. Now, phases of cell cycle. Cell cycle is divided into 4 parts, or we can say mainly 2 parts. We can say M phase and interphase. See, in the interphase, the cell duplicates its DNA, and then for it to do growth, then it does its daily chores. While in the M phase, it does growth. It does divides itself, or mitosis or meiosis. We'll learn. So these are the phases. And the interface is also divided into some parts. We'll learn here some subparts. We'll learn. And also the cell cycle completes after every 24 hours, or in some bacteria even 90 minutes. So now the cell cycle is divided into 2 basic, as I have already told you, interphase and M phase, or we can say meiosis phase or meiosis phase. So the M phase represents the place where the actual cell division occurs, or also when the cell divides, all the things are divided, it just separates itself. Now because interphase makes a lot of a part of the cell cycle. so that all the 90% time it does growth, and it duplicates its all the body parts, and the DNA, it duplicates its DNA, RNA, all the body parts, and then in the end phase it just what it does to duplicate itself, to divide itself using various processes such as prophase, amphase, anaphase, telophase, and cytokinesis. We learn more about them in detail. This all I have told you. And also, interphase, though called resting phase, has a lot of features and forms 90% of the cell cycle, as I have already told you. So let's proceed. The interphase is divided into 3 further parts. Now, as I told you, it has some subparts, so it has 3 subparts: G1, S, and G2, or which means gap 1, synthesis, and gap 2. So, in the gap 1, or the G1 corresponds to the interval between meiosis and mitosis, or means it can say that it has just divided, and then it comes to a stage of G1 where it growth recovers from that is now a daughter cell, it's another particular cell, then it grows, it does not start replicating its DNA, it does its daily work, it grows, and somewhat the DNA gets start mixing with the protein fibers, and very slowly they start thickening, or they can negligible amount of they start into the chromosomes. Chromosomes what are basically now in the mitosis or meiosis, 2 daughter cells, or we can say mitosis, 2 daughter cells, because learning about mitosis, 2 daughter cells are produced. So they must have, now suppose there is a cut, and you want to have it filled, or blood will go out, and that won't work. If you have even a minor cut, then it can cause death. So what happens that the cell have to duplicate themselves for the growth and lot of things. For example, there's a cut in your hand, and what if now, as I in earlier video I have told you that there are a lot of kinds of cells, stomach cells, and a lot of kind of cells. So suppose there's a cut in your hand and your cells start making stomach cells. That is not right thing to do at that time, because hand cells, the skin cells are needed at that time to fill that cut. So that is what mitosis does it. So as it divides into 2, now that daughter cell is another. So it grows, and it takes its position, then it grows, and it do some daily things such as eating energy and making energy. The organelles in the cell make energy, mitochondria make energy, and protein synthesis happens, and a lot of things which are of the daily cycle of the cell. And then it enters the S phase, or the synthesis phase, where now the chromosomes, now because if they make 2 daughter cells, they have to have the same kind of DNA to make same kind of cells. If you have same kind of DNA, same type of DNA, same number of DNA, then only you will make same kind of cells or clones. So now, because these 2, suppose there are 2 DNAs, and you want to make 2 daughter cells of yourself, and every daughter cell should have 2 chromosome DNA of, for suppose. Now you only have 2, and you may have to make more, which you have to divide to 2, which both of them should have 2 chromosomes. So in that way you should first of all make your chromosomes 4, so that you can divide into 2 and 2 chromosomes given to each daughter cell. The same it happens now, because there are a lot of DNAs. So first, to help them make it easy to arrange them, they come in bonds with the protein fibers, and then they condense to form X-shaped structures, thick at the bottoms, or we can say evenly thick. So these form and they thicken, and they can be seen in the S phase or the synthesis phase. Under microscope we can see these chromosomes thickening, and then comes the G2 phase. As this is already have told you, G2 phase proteins are synthesized. Now to divide it needs some energy, and then it needs some energy, because when the daughter cell starts, it cannot start making its own food. Oh, I am born, let's make food. So it takes some food for the another cell, and then for its growth and for energy. In G2 phase it does all this, and then it finishes the interphase, and then moves to the M phase, or the mitosis phase, which we will learn. You can see this amazing. So here's the G1, it duplicates its DNA, because in mitosis what happens, mitosis, that it duplicates its DNA, but that DNA is divided into two parts, so still half is gone. It's not that much duplicates its DNA. So when it divides into two daughter cells, then what happens, that it duplicates some of its DNA which is needed, and then some, because at now when it divides then two daughter cells, then it duplicates some of its DNA, and then on the cell cycle then comes the synthesis when the chromosome began to appear, and the G2 phase when they make protein and food. Let's proceed now. Now we'll see M phase. Now we just learnt about interphase, and now the 10% of the cell cycle, let's consider it as dividing. M phase, or here the mitosis phase. So now what happens, now it start dividing as all the things, the matter, the DNA are duplicated, or we can say not duplicated, just have been ready, because just have duplicated and considered it was 2, then it becomes 4 for the dividing the two daughter cells, and so it case happens, and then it comes to the M phase when it's just ready, but only the physical part is there joined, and the rest things, the DNA, the matter, are all duplicated or doubled in number. So the M phase is now made into 4 subparts: prophase, metaphase, anaphase, and telophase. This you can remember by PMAT. P-M-A-T. The first letters of all. PMAT. So now you see this. This is all just the stages of meiosis. Meiosis in just a snap of time. You can just see this GIF. Then I'll explain you all the parts, what happens in all these phases better. So you please see this. You can see this. You may not have understand it. Now let's understand it. Now the first stage, prophase. Now in the prophase, what happens, that in mitosis, what happens in the prophase, see here, in the prophase, now that's nucleus. If you have seen that earlier video, I have told that it has some nucleus membrane. It starts to disappear, and now that main thing which I didn't told you at that time meaningfully, that can you see this spindle fibers? These also duplicate or double at the time of when the DNA is duplicating, and they come to the opposite ends, and this nucleus, that covering nucleus, and it disappears. Can you see that this nucleus disappeared, that blue pink colored covering disappeared, that disappears, and these spindle fibers come at the opposite poles, and as the chromosomes are quite visible. You can see this all, which I have told you. Just I need to tell you that this structure of a chromosome. Now the structure is a chromosome is like an X-type structure, which is 2 sister chromatids, and it is joined at the center through a centromere, a circle, a gluey circle which joins the 2 sister chromatids. Now what are chromatids? Oh, these sister means these are just like same, just they are full of DNA and wrapped in protein. Protein then is wrapped with DNAs and tightly packed, and it condenses in hordes, and this becomes one, and the centromere, and then comes the another sister chromatid, and they become one chromosome, and we count the chromosomes through the centromere. So 4 sister chromatids, and we'll count the centromere, which we can say they are 2 chromosomes. And with all these I have told you. If you want the PPT, it's in the description box below, and rest these things are not important. Now metaphase. Prophase, you can remember from the pro that it was early. Pro means early, first. It is first. and then comes metaphase. Now what happens in metaphase? Then see in the GF, now these spindle fibers, these grow and grow long and attach the centromeres, and they took hold of the sister chromatids. They hold them, and these spindle fibers come out and they hold them and they align in the middle, because they are floating, the chromosomes are floating like this, and then the spindles come and attach them and they adjust their position so that these all can come in the middle of the cell. As you can see, M metaphase, M for middle. So this you can remember, chromosomes arrange themselves in the middle, or it's a metaphase, or they arrange themselves at the equator. I hope you can see the GIF. Anaphase. Now what happens in the anaphase? Now they are arranged in the middle. Now A for away. Now the sister chromatids are pulled away. Oh, you should not separate sisters. But if they'll not separate, then how will human work? So their sister chromatids divide. Their sister chromatids are pulled. Now the spindle fibers contract. They get short, and as they've attached them to the spindle chromatids, they tear apart and they go with these spindle fibers. A for away, and in anaphase, these all go apart. All I have told you, they get at the opposite poles close to the spindle fibers. Now telophase. Telophase, which happens that the nuclear fiber, you can remember telophase for 2, tel for 2, T for 2. So this the nuclear membrane and the nucleus comes again, and these chromosomes start to get back into the DNA. The DNA get unwrapped from the protein fibers. The protein has this circle-like thing, and they get unwrapped, and the chromosomes begin to disappear, and the centromeres get aligned, because they were 2 centromeres, so they get near the nucleus. Each one of them get near the nucleus. Telophase for 2, and they get 2 nucleus. So just there they are attached to their cytoplasm. Rest all this divided. All they just Golgi protection, all the parts come back. Now cytokinesis. Now as we know that at the telophase, only that cytoplasm was left. So now you can remember, see, for cytoplasm, this cytoplasm disappears, this cytoplasm shortens and divides these two into daughter cell, a mother cell into 2 daughter cells, and now this unwrap, and though the daughter cells have the same amount of DNA required, so from one cell we can get 2 cells, and it goes on, other cells also duplicate, so that they can make many million cells, and which can fill that cut or for our growth. And this all I have told you. Just if you want to see, you can see this GIF. Cytokinesis, the gap deepens, and then until they are separated. Now let's move. So this was all in this video. If you like this video, please like and subscribe my channel, and don't forget to click the bell icon below to get regular updates of my videos. And also don't forget to see the next part, meiosis. And I remind you again to stay curious.